Okay, good morning everybody, ladies and gentlemen. Thank you very much, the organisers. And I think Dr. Rao confessed that he was behind me being here, so thank you very much. Like him, I'm also a little bit agitated by this subject. It's a very interesting one. So, after his presentation, well, two things. First of all, I would have written a completely different presentation, so that's one thing. And the second thing is, I'm really glad he's agitated, because we can be agitated together. Which is good, because this is a subject where I think we're all trying to learn a lot about how to do things, or how to work with this particular topic. Maybe better than we've worked with similar topics in the past, I don't know, but I think that's our hope and expectation. So, I'm going to try and tell you a few things about OECD and some of the things that have been happening there on this topic, and some of the things that I've been thinking about a little bit. I'm very lucky because I have another presentation tomorrow. So if this one doesn't work too well, I can come back again and try again tomorrow in the morning. It's a different subject, but that won't stop me. So just a few words about OECD. I think, unbelievable this works, good. About OECD, we are an intergovernmental organisation, so our job is to work together with governments, and the primary group that we work with on biotechnology is called our Working Group on Harmonisation of Regulatory Oversight in Biotechnology. This is a group that's composed of delegates nominated by governments, normally from those ministries or agencies that are responsible for regulating products of biotechnology. This group has been around since the early 1990s, and there was work before at OECD on the same topic, but this working group has been around since 1994. It deals with emerging issues related to harmonisation. We discuss new products and techniques that are used to produce them. Recently we've been thinking about new traits, such as stress tolerance. We've been thinking about new plant breeding techniques, at least since the beginning of this year, when in February, as Dr. Rao mentioned, we had a workshop in Paris. We think about things like data, how data that's used in risk assessment can be shared amongst regulatory agencies, and we particularly produce things like, or things that are called consensus documents, which might come out of this work. And those consensus documents are what our governments believe is important information that can be used in risk assessment and shared between governments. And that will mainly be the topic of my presentation tomorrow, so I can kind of fit all of this a little bit into perspective. Just a couple of things. OECD has 34 member states, but in particular in the work that I do, and in particular in the working group, we have had a long history of working with countries that are not members of the OECD, and that includes India, and it includes countries like Brazil and Argentina and South Africa and China, and I'm going to miss some, but countries with which biotechnology, and especially biotechnology related to agriculture, is an important feature of their economies. So when I talk about OECD, it's not just our governments, it's quite a number of others besides. Okay, so it was about sometime last year we began to think about this issue of new plant breeding techniques, and we had a workshop in February, as I said, and we also had a questionnaire that was circulated amongst governments in advance to try and get their attitudes or their responses to what is happening in their particular countries vis-à-vis new plant breeding techniques. And we had quite a lot of responses. We haven't published anything yet, and there's a reason for that. Because we are an intergovernmental organisation, we wouldn't be a proper intergovernmental organisation if we didn't have all sorts of fascinating bureaucracy. And whenever we put material together, part of the bureaucracy involves us going back and consulting with countries and having a consensus on what's published, and that can take quite a while. So I can't share with you, well, I can share with Dr. Rao, and he can do with it what he will, but we haven't actually published any information yet. So I just want to give you a little bit of a quick summary about what we have discovered so far. As I say, information gathering, we had the workshop and we had a questionnaire. Just to go into a little bit of detail, we're trying to understand the types of products that are being developed using these types of techniques. We're trying to understand the techniques. By understand the techniques, I mean understand the molecular basis behind the techniques, because that's very important because what we're talking about here is technical information related to the science, but we're also interested in the consequences from a regulatory perspective. What happens when we get products emerging from the use of these technologies, and what a regulator is going to do with them. And that's the kind of link between the technical information that we're talking about today, mainly the science, the molecular biology, and tomorrow we have the regulatory part of it. But these two things go together, which gives me an excuse to talk about both a little bit, but neither in huge amounts of detail. One thing we were very interested to know, and we are still interested to know, this is all moving very quickly, is the practical experiences countries may or may not have had with environmental risk assessment of the products developed using these types of techniques. Do we know of any new safety issues that are specific to these types of products that haven't been thought about before when we're talking about traditional transgenesis, for example? Is there anything new? Are there any differences in approaches to environmental risk assessment between countries? Because if there are going to be differences, then, as Dr. Rao mentioned earlier, if there are differences, then there could be problems in trading these types of in the commodities that are developed using these types of techniques. And that is not a very good thing to happen if it can be avoided, to have major trading issues. And then at the end of it all, what are we going to do as next steps, if anything, at OECD and in our working group? By the way, I'll just give you at least a couple of ideas here from these points on this slide. Well, I'll talk more about each of them, but one of the main things when we're thinking about environmental risk assessment and if there are any new safety issues, perhaps one of the most important things I can say immediately is that no one that I know of has yet come up with a new environment, with a new risk assessment type of issue, a new environmental risk assessment type of issue that we haven't thought about before, associated with other, say with traditional transgenesis or with other types of technologies. So we can all be very relieved to know that there's nothing really new that we have to think about. That may not persist, but that's certainly the case at the moment. We actually focused in our workshop earlier on these techniques. My original plan was to give you a little, I've got some nice little diagrams that me and my colleagues have invented, which give a quick summary of each of these techniques. And then I suddenly realised there's going to be a lot of detail coming after I finish speaking about many of these things, and so I'm not going to dwell on the scientific molecular biology of these, but these are the types of techniques we particularly focused on in our discussions at OECD. Oh, by the way, yeah, maybe I should have put this earlier. These are the countries who responded to the questionnaire and our request for background information. And these are the main sources of information. I'll give just a few quick summary comments about what we learned as we went along. But these are the countries that we heard from. It was a relatively small workshop that we had with 130 participants from governments mainly and other intergovernmental organisations. We based this list here comes from the work of Lucer et al. And the reason why we use this is because it was published in the open literature, which is a good start. And also it had a couple of elements to it that we were interested in. One is the molecular biology, the other is also issues, questions around the potential regulatory status of these things. Sorry, I'm going the wrong way. So the overview of the techniques that we began with was from that particular paper. We had a number of presentations, overviews from people involved in research using these techniques and people involved in the development of products. We had academic, the public sector perspective, we had the corporate perspective, and we had some more detailed presentations and discussions from countries plus the EU on what they are doing in this area. Okay, in terms of environmental risk assessment and safety assessment, we wanted to have more insights into what people had on that particular topic, and in particular related to the types of plants. We're mainly talking about crop plants here, of course, and into the type of plants in development. And as I said a little bit earlier, the environmental risk assessment was something that was of particular interest. And as I say, we haven't as yet discovered any new issues that we are not familiar with so far. One of the questions related to the types of techniques that's being developed in countries, and is the list that we were starting with relevant? Are there additional techniques? And in particular, nothing surprising here, the techniques that people were interested in and spoke about beyond the 7 that I mentioned earlier included accelerated breeding, RNA interference, technologies and CRISPR-Cas types of technologies. That's the original list on the side there that we began with, and those other things were added to the to-do list, as it were, what other techniques we might need to think about. When we asked about research or product development that was underway, these are the most mentioned techniques at the top of the list: cisgenesis, intragenesis, ODMs, and SDM applications. I know from the conference program we're going to hear quite a bit more about these. The most mentioned crops, and these are from the 21 countries making responses, were apple, potato, maize. The most mentioned traits were fungal resistance and herbicide tolerance. Nevertheless, most developments that are underway are still in the research phase. That's actually very good news for you, Dr. Rao, because it means you're right at the front of all of this. To look into this issue and attempts to formulate recommendations, but most countries do not yet have practical experience with risk assessment. A few countries said that they did. Maybe I'll come back to that a little bit. And many countries, at least at that stage, referred to the EFSA opinions that have been worked on. And I know we will hear more about that tomorrow, I think. So, although these are not EU member states, some of our countries are EU member states, but not exclusively, nevertheless, these types of opinions seem to have had an influence on people and what they're thinking, whether or not they're an EU member state. These are just some of the examples of the countries that mentioned whether or not they have experience. It's always good when you do a survey like this. We cannot force governments to reply to our questionnaires, but it's always good to have negative responses as well as positive responses. So people say, well, we don't have any experience, but still this is what we think. And that's always useful. Otherwise, it's a very kind of biased type of sample. A lot of countries refer to various position papers, plant breeding associations. Some countries mentioned the fact that they've already had or are anticipating requests from industrial developments of these types of products, particularly related to what might be required in an environmental risk assessment. And we had mention of, not surprising, a lot of people stressed the importance of a science-based environmental risk assessment, and there was a little bit of a debate in the responses we had about product-based approaches, process triggers for regulatory assessment. This other question that we looked into was really asking countries what they thought was important in their particular circumstances when thinking about these techniques. And top of the list was really questions or issues related to the regulatory legal status of NPBTs. There was other people thinking about, can we not be more proactive in the future in some way of recognizing new types of emerging technologies in the future, so we can take a more anticipatory approach. The third is a little bit of a more technical issue, but it relates to the fact that a lot of these techniques you are unable to detect the end product at the end of the process. So you can have, if you regulate these things, you cannot determine whether or not they've been used in the end product. And where does that leave us if we don't have detection techniques? And a number of countries said they were also interested in the development of animals, particularly agricultural animals, with these new types of techniques. Now, this is really tentative and speculative, and these are the ideas of me and a couple of colleagues. And the reason I put these in here is because we were trying to struggle a little bit in how we bring these things together, the science side of things and then the regulatory side of things. So please consider this as something, as thinking in process. But what we tried to do is come up with categories of NBTs. We were trying to think how what the regulatory response might be. So here we're talking about a category of NPBTs, which are basically mutational types of technologies, specific mutations to a targeted site in the genome. And down the left-hand column you will see the types of techniques which are probably the most common techniques in real practice at the moment. What we were trying to think of is these are basically mutations, and in many regulatory systems there are one or two exceptions, but mutational type of breeding is not normally a regulated process. And these are mutational types of technologies, very site-directed and all the rest of it, but normally exempt from a regulatory system. So that's already a category where you could imagine, with a bit of luck, they would not be regulated. These as I say, this is just pure invention. Then we thought of another category, end product free of transgenes or transgenic material. And these are really the three techniques we came up with here. And this comes back, this is also from a regulatory perspective, the thought there's no transgenic material in there, and or material derived from transgenic material. Might this be also something you would not be interested in if you were a regulator? This is maybe not as hard and fast a conclusion as you might come to with mutational breeding, but still, if you have no transgenic material there, why would you regulate it? It's just a question. I'm sure people can invent all sorts of reasons, but okay. Normally, in most regulatory systems, we are not that much interested in regulating products where it's been derived from relatives. That is explicitly excluded in some regulatory systems. So cisgenesis and intragenesis, which we will hear more about later, if we're dealing with material that is only material from a sexually compatible relative, is that something we should be interested in from a regulatory point of view? I know there are all sorts of flaws with these categories, and I know that if you are using agrobacterium or other similar techniques to transform a particular cell, or whatever it happens to be, often you have material derived from non-compatible relatives. So, I don't know, maybe you can think of some other categories. But this is the category we I'm just trying to find a way of thinking about how we match up these types of techniques with the regulatory requirements that we have. And finally, well, two very different, but with the similarity that the end result may be material if you're transforming only a particular part of the plant tissue, and the product that is being used or consumed or whatever afterwards doesn't contain any transgenic material or products derived from transgenic material. Is that another thing? Is that something else where there might be some kind of regulatory exemption or whatever it is you would want to call it? I do understand, once again, there are flaws with this type of thinking. If you have, for example, the grafting on GM rootstock, excuse me, material can pass from the rootstock to the shield, but so there are flaws, but that's another category. Am I okay? Yeah. Sorry? They are, thank you. I thought you were pressing me for time. Okay, so those are some really speculative things. Sorry? Those are just some speculations, but all we were trying to do, and this is just me and a couple of our colleagues, is try and think in some kind of logical way the science and what might be the regular consequences. So I'm just sharing this. If you find problems with the categories, I think that's at least some kind of for us to think about. This is another very, this is a different type of slide, but this is just an example that we know of three sort of regulatory decisions that have been taken around three particular types of products. The first two are basically mutational type of technologies. That would be my category 1, if you like. And in the first case, approval for this herbicide canola variety in Canada, and the USDA APHIS, that's the USDA, the United States Department of Agriculture Animal Plant Health Inspection Service, have said that given the system, this like classical mutagenesis. This is published as a letter on the USDA website. And this looks to them like so they are not going to regulate it. That's US who are responsible for commercial planting, deregulation. Anything about the decisions that FDA or may make vis-a-vis. The second, also from my category 1, and once again it's a zinc finger maize, and once again USDA APHIS has said that they do not regard this as a regulated article. The third one is a system. That's my category 2, 3. And this is from Wageningen. I think we hear more from Wageningen people tomorrow. Oh yes, right, follow me. And what the USDA is saying there is they regard it as a regulated article. This is because it has involved the use of agrobacteria. And part of the basis of USDA APHIS regulatory decision, a plant pest or a plant pathogen has been used in a process, then it is regulated. And that's the case here through agrobacteria. So there have been some regulatory examples and go on what decisions people might take. I admit not very much, but this is moving forward. As more of these decisions are taken, we're thinking about the basis on which something has not been regulated or something has been. And it's even possible that the tentative categories I mentioned may be a thinking around how to move along. That's all from me for the moment, but I'll get back tomorrow to you. Other things to say. This is my team. One person here who has left since. I had a guy working with me from the Netherlands called War. He has left my team now to actually go and work in FAO, which is already interesting. But he did a lot of work with us on this also, and I should have had his name here. Thank you.